Oh, very good morning, friends. Welcome to my channel. So today we'll discuss ICH Q8 Pharmaceutical Development Guideline. For the sake of making video, I divided this video in the two parts. So this is the first part of the video in which we discuss ICH Q8 Pharmaceutical Development. So friend, as we know, this channel I created for helping the pharma freshers and anybody who wants to be a part of this pharma learning journey. So please subscribe the channel so that you will get the regular updates of the video. And if you like the video, then please like and share also so that it will reach to the maximum number of peoples. So let's start the video. So friends, in both videos, we learn about the general consideration of pharmaceutical development. We learn about the new approach and conventional approach of pharmaceutical development, design space, real-time release testing, what is the control strategy. We learn all these things by the examples and we'll conclude everything. So friends, the aim of the pharmaceutical development is to design a quality product and its manufacturing process to consistently deliver the intended performance of the product. So sometime question came, क्यों नहीं हम directly scale up batch ले लेते हैं या क्यों नहीं हम directly exhibit batch या commercial batch ले लेते हैं? तो इसलिए क्योंकि if we do directly without any experience, without understanding the product, then chances of failure will be more, right? This product development is a journey. So we cannot go directly to the scale up and execute batch. First we need to understand the product, so that when we'll go through that journey, then only we can design a quality product with a robust manufacturing process, so that that product consistently deliver the intended performance which is expected from that product, right? So one more thing, that today what the current perspective is that quality cannot be tested into the product. That is, quality should be built in by design. What it means that first you take a batch, then you wait for the result, whether it will be pass or whether it will be fail. So this is the one approach, right? But the best approach is that you made that much robust product. It is that much tested, and it is developed in such a way that the design itself inbuilt the quality, that if you manufacture the product with that range parameters, then quality will be assured offshore. Means there is no requirement of testing also. The quality is present within the product because of its design. So that is the current approach. Information from pharmaceutical development study can be basis for quality risk management. So whenever we go through the journey of the product development, so whatever information we get in the development, right? What are the critical material attributes, what are the critical process parameters, right? And what are the CQAs of this product, right? And what is the relationship between the critical material attributes, formulation and process variables, and critical process parameters. By understanding that, we can that understanding will provide a basis for our quality risk management in future in the life cycle management of the product एक बार स्केल अप बनने के बाद जब कमर्शियल स्टार्ट होता है, तो हमारा जो थ्रू आउट अंडरस्टैंडिंग है प्रोडक्ट का, जो डेवलपमेंट का अंडरस्टैंडिंग है, जो स्केल अप का अंडरस्टैंडिंग है, जो एग्जिट बैचेस का अंडरस्टैंडिंग है, और जो आपका एक रिलेशन है उसके मटेरियल एट्रीब्यूट, जो एक्सीपिएंट या एपीआई जो उनके मटेरियल एट्रीब्यूट्स, और जो आपके प्रोसेस इन्वॉल्व हुआ उसके क्रिटिकल प्रोसेस पैरामीटर्स में, और जो क्वालिटी एट्रीब्यूट्स है प्रोडक्ट की, उनके बीच का जो रिलेशनशिप है, राइट, वो हमको हेल्प करेगा, उसकी अंडरस्टैंडिंग हमको हेल्प करेगी फ्यूचर में क्वालिटी रिस्क मैनेजमेंट में. Okay, so this product can be developed by the minimum approach and by also the enhanced knowledge approach, which we also say that QBD approach. What it is, we'll discuss later in the slides. So friend, structure of the parent guideline is consist of pharmaceutical development, which will have an introduction, what is the reference product, what is our product, what is our approach, what are our batch sizes, etc. Then components of the drug product. So drug product mainly consist of drug substance and excipient. What are the critical material attributes of the drug substance, what are the critical material attributes of the excipients. Then drug product development. So in the drug product, it involves pharmaceutical formulation development. In the formulation development, we explain what are the trials we have taken, how we conclude our prototype formula, how we optimize our formula, and what is the final formula. Then overages, any overages proposed in the formulation. So there is a difference between the overages and process loss. For example, in capsule filling, we know that empty capsule cell required more because of the process loss. So that is a process loss, that is not a overages. Whenever we are taking some additional API because of some degradation or because of any other reason, then that is the overages which we have to disclose in this section. Then physicochemical and biological properties, manufacturing process development, which will explain the what is how we develop our process, how we optimize our process, what are our critical process parameters. Container closure system, which will give an insight of the packaging development, microbiological attributes, compatibility, and glossary. So, friend, as we discussed that pharmaceutical development can be done by the two approach. First is the minimum approach. Second is the enhanced approach. We will discuss at the end of this video also the comparison between the minimal approach and the enhanced approach or QBD approach. But minimal approach is also consist of, in all cases, means in minimal approach and as well as in the enhanced approach. In all cases, sufficient development has to be done so that a product can be released to the market. So in minimal approach also, you have to identify quality target product profile. Means what product you want to make. Then you have to identify critical quality attributes of the drug product. What critical quality parameters you require in your drug product. What are the critical quality attributes of the drug product? These all terms, quality target product profile, critical quality attributes, will discuss in this video. Determine the quality attributes of the static material. What are the critical material attributes of the drug substance? For example, API, its PSD is important or its polymorphic form is important. So those critical material attributes will determine in the minimal approach also. In the excipient also, PSD is important, and every excipient has its critical material attributes. So that will identify in the minimal approach also. Then based on the our target, based on our CQAs of the drug product, and based on the CMA of our drug substance and excipients, we will select appropriate manufacturing process. We will define that manufacturing process, and based on that we will show also how we are going to control our product. Means how, what is our control strategy, how we are going to control our drug substance, our excipient, material attributes, our critical process parameters, and how we can ensure that our final drug product is meeting its quality attributes. So this all activity should be performed in the minimal approach also. So friends, in the enhanced approach, we do a systematic evaluation by understanding and refining the formulation and manufacturing process. In the enhanced approach also, we will identify the material attributes, we'll identify the critical quality, we'll define our critical quality attributes of the drug product, we will define our quality target product profile. In addition to that, we will identify the material attributes and process parameters that can have an effect on the product CQAs. We will determine the functional relationship that can link material attributes and process parameter to the product CQAs. So in addition, we understand the interaction, the link between the critical material attributes, critical process parameter to with the drug product CQAs. So that is the difference between the minimal approach and the enhanced approach. In enhanced approach, we do all things which is required in the minimal approach also, but in minimal approach we are not studying we not able to find the perfect relationship between the critical material attributes, critical process parameter, and our critical quality attributes. So in the enhanced approach, we understand individual impact of our critical material attributes or critical process parameter on the CQ of the drug product, as well as we understand the interaction, the relationship between, we understand the combined effect of the critical material attributes and critical process parameter on the drug product. So that relationship, that understanding will help us to develop a more robust process, to develop a more robust product. So based on this relationship, based on this understanding, we can establish more appropriate, more robust quality control strategy in the enhanced approach. So friend, what are the benefits of the enhanced approach, enhanced development approach? We will discuss in this slide. So depending on the level of the development achieved, means depending upon the scientific understanding of the development, scientific understanding of the product, and an adapted quality system in place, opportunities exist to develop more flexible regulatory approaches. For example, risk-based regulatory decision, reviews and inspections. So, if we have established a relationship between the our critical material attributes, our critical process parameters, and the impact of the material attributes and process parameter on the critical quality attributes, if we defined it properly by the mathematical understanding, then if any deviations happens, any problem comes during the commercial manufacturing, then it can be defined, it can be understand, that issue, that concern can be understand based on the information, scientific understanding available by the development, by the development approach, as well as any risk based regulatory decision can be understand and can be justified based on that scientific understanding. Then manufacturing process improvement within the approved design space described in the dossier without any regulatory review. If we defined our design space properly, correct. If we defined our design space properly, then for any process improvement, if we are changing within the design space, then it is not considered as a change. So it will help for the regulatory review, and it will justify our we can back our design space, we can back our development studies, we can back our approaches by the scientific understanding to the reviewer, and it will be much easier for the reviewer to rely on that, to it will much easier for us also to convince reviewer. Then it will reduce post approval submission. Then real time release testing, leading to reduction of end product release testing. So nowadays we are talking about the pad tools, nowadays we are talking about the real time release testing, and by the enhanced approach, we are ensuring our relationship between the parameters and its impact on the critical quality attributes. So the end time testing reduced end time testing is reduced, and by NIR approach, by different pad tools available, we ensure our quality within the process. We ensure our real time, we can do the real time testing, which will reduce the end product release testing. So these are some benefits of the enhanced development approach. So, friend, in this slide, we will understand the basic term QTPP, quality target product profile. So, in the generic industry, we have a reference product. Generally, our target is fixed. So, in QTPP, we are enlisting what is our target. So, first, before starting of the development, first we should understand what is our target. Correct. Our target will be derived from the our reference product, right? So the quality target profile forms the basis of design of the development of the product. Consideration for the quality target product profile could include intended use in the clinical setting, route of administration, dosage form, delivery system, dosage strength, container closure systems, therapeutic moiety release or delivery, and attributes affecting pharmacokinetic characteristic. Example: dissolution, aerodynamic performance, appropriate to the drug product, dosage form being developed, drug product quality criteria. Example: sterility, purity, stability, and drug release, appropriate for intended marketed product. So based on our reference drug product, we will define our quality target product profile, what route of administration we want, what is our dosage form, what is our delivery system, it is IR, it is ER, what is our dosage strength we are developing, what is our packaging system, what is our pharmacokinetic property, what pharmacokinetic property we want from our drug product. So what are the quality drug product quality criteria, what is our dissolution assay, these all will define the quality target product profile. Hope you are able to see this table. So this is our QTPP table. So this I taken from the QBDIR example. So we should enlist our QTPP as per this table. Like dosage form, it is tablet. Dosage design, it is IR tablet, right? Route of administration, it is oral. Dosage strength, it is what is our target dosage strength will mention here, right? Now what is our pharmacokinetic consideration, right? So we want a bioequivalent to the RLD. What Tmax we want, we can mention in our QTP target QTPP. What is our stability? As per our RLD, we can define our shelf life. So what our drug product quality attributes? So what quality attributes we want in the drug product? Like what physical attributes we want? What identification we want to comply identification? We want to comply assay. we want to comply content uniformity, dissolution, degradation product, residual solvent, water content, microbial limits. So whatever the critical quality attributes of the drug product, we can enlist there. Then what is the packaging? What is the packaging we are targeting? So any labeling consideration, like here written similar food effect as RLD. So any labeling, special administration, or any labeling we want to mention, we can enlist it in the quality target product profile. So this is a way how we will enlist our quality target product profile based on the RLD, based on our target. First step in the development is to define your quality target product profile. So first we'll identify our RLD. After identifying our RLD, we'll define what is our target. We define our target by the QTPP. Now, friends, after defining our QTPP, we need to understand what is our CQAs, what is our critical quality attributes. So, a CQA, what is a CQA? A CQA is a physical, chemical, biological, or microbiological property or characteristic that should be within an appropriate limit, range, or distribution to ensure the desired product quality. CQAs are generally associated with the drug substance, excipients, intermediates, in process materials, and drug products. So this is the definition of the CQA. CQAs of solid oral dosage forms are typically those aspects affecting product purity, strength, drug release, and stability. So CQAs for other delivery system can additionally include more product specific aspects such as aerodynamic properties of inhaled products, sterility for parenterals, and additional property for the transdermal patches. For drug substance, raw materials, and intermediates, the sequence can additionally include those property, example particle size distribution, bulk density, the effect of the drug product sequence. So here we are talking about the drug product sequence also, as well as our critical material attributes of the excipients and drug substance also. Potential drug product sequence derived from the quality target product profile and or prior knowledge are used to guide the product and process development. The list of potential sequences can be modified when the formulation and manufacturing process are selected as product knowledge and process understanding increase. Quality risk management can be used to prioritize the list of potential sequences for subsequent evaluation. Relevant sequence can be identified by an iterative process of quality risk management and experimentation that assess the extent to which their variation can have an impact on the quality of the drug product. First, we have to identify all quality attributes. What are the quality attributes of the drug products, right? After identifying the all quality attributes, we have to enlist what are the critical quality attributes, correct? So those critical quality attributes will be assessed. The impact of the material attribute means एपीआई का प्रॉपर्टी, एपीआई का प्रॉपर्टी जैसे आपको मैंने बताया पार्टिकल साइज, एक्सीपेंट का कोई प्रॉपर्टी है कि जैसे स्वेलिंग इंडेक्स है फॉर इन द केस ऑफ द डिसइंटीग्रेंट, और पार्टिकल साइज है एक्सीपेंट्स का, जो भी क्रिटिकल मटेरियल प्रॉपर्टी है इंडिविजुअल एक्सीपेंट का, उसका रिलेशन ड्रग प्रोडक्ट के सीक्यू पे कैसा है, वही हम डेवलपमेंट में स्टडी करते हैं. तो वो कौन कौन से सीक्यू होने चाहिए जो हम स्टडी करेंगे. सो हाउ टू आइडेंटिफाई दैट? First we have to understand what are the total the quality attributes of the drug product. For example, in the last slide we have seen that table. So based on that we'll enlist our quality attributes. In next slide you will see, and then we have to see that what are our the critical quality attributes. So let's move to the next slide. So friend, this is the CQA table, which again I am taken from the QBD example. So here we have enlisted all our quality attributes, and we are identifying which is the critical quality attributes of which is not the critical quality attributes. For example, the physical attributes. Physical attributes, appearance, order, size, score. It is for a tablet formulation. So appearance is not linked to the patient safety, but yes, it is linked to the patient acceptability. So it is a quality attribute but not a critical quality attribute. So generally we fix our target and will ensure our target. It is not required to study in every batch. So it is a quality attribute but not a CQA. Order, it is a quality attribute but not a CQA. We should ensure, but need not to study in all batches. Size, we have a size shape guideline. We also made one video on that guideline also. So size is also, if it is above that, it is nearby to that guideline, that is 17 mm, something like that. If that is a specific case, then it is a critical quality attribute. Otherwise it is a quality attribute, not a critical quality attribute. Scoring, if scoring is present, then we have to comply as per guideline, then it will become a critical quality attribute. But if it is unscored, then it is not a critical quality attribute. Friability, same as like that. So we have to identify based on the product to product case, based on our QTPP, we have to identify which quality attributes is critical, which we want to study in our entire development approach, right? So, like identification. Identification means whether the drug we are intended, that is present or not present, right? So, it is a critical quality attribute, but this is critical quality attributes, but it is not required to study in all batches, right? Because it can be easily controlled by our quality management system. Means it is controlled by our specification. We can ensure in the API rules also by identification, in the drug product rules also by identification. So it is not a critical quality attribute, right? But assay, yes, assay can be affected by our process, right? So if our process have some variation, if by that assay can be impacted, so yes, it is a critical quality attributes. Content uniformity can be affected by our process, right? Our process design, so it is a critical quality attribute. Dissolution will ensure our viability, right? If our dissolution will ensure our viability, so it is a critical quality attributes. Yes, it is a critical quality attributes. So by the help of this type of table, first we'll define our QTPP, then we will define our critical quality attributes by the help of this type of table. So these are the additional quality attributes, and of this again I am taken from the this QBDI example only. So degradation product, impurities, this is a critical quality attributes we will study during the development. Residue solvent, if you it is also critical quality attributes, but if you are we also made one video how to as per ICS Q3C, you can refer that video. So if you are using residual solvent and if you feel that it is critical quality attributes in your product, then yes, it is, you have to study. If you can define based on that guideline, then it is need not to study, only in final product you can ensure. So water content, same way you have to if your product is sensitive to the water moisture, and there are chance if it is hygroscopic, then you can consider it as a critical quality attributes, or it is simple quality attributes. Then microbial limits, in case of the liquids it will be critical, in case of solid there are less chances, so you can ensure in the final end product testing also. So this way you have to enlist your all quality attributes, and you have to understand what are the critical for your product, right? So it will depend upon the product to product, right? So based on that you have to identify your critical quality attributes, and those critical quality attributes, then after defining the critical quality attributes, we have to understand the impact of the material attributes on the like API, impact of the API material attributes on these CQAs, then impact of the formulation variables on these CQAs, and then the impact of the process variables on these CQAs. And after understanding all the variables on the CQAs, we have to what is we have to define based on our target. We have to come over the design space, and then we have to define our control strategy. So this is the cycle or chain of the development. So friends, we understand that first we have to identify our RLD, then we have to define our QTPP, then we have to identify our CQA. After that, we have to identify our CPP, critical process parameter. So what is the critical process parameter? We'll understand in this slide. And what is a development? So now we can understand what is a development. Development is a linking of material attributes and the process parameter to the drug product CQAs. So if we that is only the main objective of the enhanced approach also. So how our material attributes and our process parameters and our formulation variables are impacting our drug product CQA, that only we understand in the development. So critical process parameter, a process parameter whose variability has an impact on a critical quality attributes, and therefore should be monitored or controlled to ensure the process produces the desired quality. So we know we based on literature search, based on our experience, based on the drug substance property, and based on the intended doses form required, we defined our process. We can select our process. For example, if we are making a solid oral doses form, if we are making a IR tablet, we can either go by a direct compression approach, we can go by a dry granulation approach, or we can go by a wet granulation approach. In wet granulation approach also we can go by a high shear granulation or by a FBP or some other process, right? So for example, if we selected the wet granulation approach, so in the wet granulation approach, what will be the critical process parameter, whether binder addition or our total granulation time needing time will be a critical process parameter, right? So we have to identify based on the selected process. We have to assess each individual unit operation, and in the each individual unit operation which is the critical operation, and in the critical operation which are the critical process parameter which can affect the quality of the drug product, which can affect our CQAs, that we have to identify, and then we have to study, and based on the study we have to develop a mathematical relationship between those parameters, as well as our formulation variables. We have to make a mathematical relationship which will define the impact of those parameter on our drug product CQAs, correct? So CPP, critical process parameter, this is the definition. Then risk assessment is a valuable science-based approach used in the quality risk management. In ICH Q9 it is defined that can aid in identifying which material attributes and process parameter potentially have an effect on the drug product CQAs. Risk assessment is typically performed early in the pharmaceutical development process and is repeated as more information becomes available and greater knowledge obtained. So we can to identify, see, we can define our process parameter, right? But which process parameter is critical, that we can identify by the risk assessment approach. So there are some tools which are defined in the ICH Q9. Just briefly we will touch those tools in our this video. So based on the help of those tools we can define which process parameter is critical. So risk assessment tools can be used to identify and rank parameters, example process equipment input variables, with potential to have an impact on the product quality based on prior knowledge and initial experimental data. Based on the initial experimental data and based on the prior experience, we can use the risk assessment tool to define our critical process parameters. So friends, there are, as we discussed, there are risk assessment tool which we will in detail will discuss in the ICH Q9 video. But briefly, I'm touching those tools. So this is the fishbone diagram. Fishbone diagram will help us to enlist all our man, material, or environment variables. So it will help us to enlist our all variables, and it will help us to go to the root cause of the concern. So this is widely used in the industry to identify the root cause of the problem. So here in the development we can use it to enlist all our variables, and then by using other tools we can identify which is the critical variable. Okay, so this is an fishbone diagram where we are showing the all variables involved in the manufacturing of the solid oral doses from tablet. So here we can place the raw material variables like drug substance. So drug substance PSD is a variable, so PSD, drug substance LOD, correct. Then aging of the drug substance, then excipient, we can mention the critical material attributes, for example diluent, we can mention the particle size and LOD. And other critical excipient like lubricant, we can mention the specific surface area. For disintegrant we can mention the swelling index. For the binder we can mention the viscosity. So all material attributes we can mention here. Then the critical process parameter, like for example in the granulation, whatever the critical process parameter we can mention. In the compression, whatever the critical process parameter we can mention. In the drying, what are the critical process parameter we can mention. Then these are the material and process variables. Then the man variables, like what is the qualification regarding operator, the variable, whatever the variability is there, that we can mention. What are the temperature and RH factor, that we can mention. What are the analytical variables, that we can mention. So all variable which are involved in each unit operation we can mention. So it will give us more enhanced view of the all variables. So after enlisting all variables, we can go to the root cause by identifying by identifying the critical variables, by nullifying the remaining non-critical variables, or where we have data or we are within range. So by nullifying that, so this is used for the problem solving approach. And in the development, we can use it to define our the critical process parameters. We can enlist first what are the critical process parameters, what are the process parameters, and then we can by using other tools we can identify what are the critical process parameters. So friends, these are the two approaches which we can use to define criticality of our variables. So this is the matrixing approach. In the matrixing approach, we one side we enlist our critical quality attributes of the drug product. At the second side, we enlist the variables, whether it is a material attribute variable, drug substance variable, whether it is a formulation variable, or whether it is a process variable. We enlist all those variable at this side. Then we'll understand what will be the impact of that variable on the critical quality attributes. If there is no relationship between them, then it is a low risk. If it is if probability is very less but it may impact the CQA, then medium risk, and if high, then it may high risk. And based on this based on by using this matrixing approach, we we prioritize our we will assess the risk assessment. We will do a risk assessment and we will identify what are the high risk variables. Those high risk variable will be studied during the development, and based on the development conclusion, we de-risk those variables, and we define our ranges, our operation ranges. So this is the way, by matrixing way, by which we define our high risk variables. We study the variable and we identify the risk of the variable on the drug product CQA, and based on the risk given, we study those variable, and finally we de-risk those variables. So this is the matrixing approach. One other approach is the FEMA, where we assign a risk priority number, and if the risk priority number is high, then that variable is high risk variable and will study during the development. And if the RPN number is less, then no need to study that variable, right? So in that risk priority number defined by understanding what is the impact. If impact is more, then we give a high number. If impact is less to affect the drug product CQA, then we give a low number. Then what is the probability of occurring that variability? If the probability is high, we'll give high number. If probability is less, we give low number. What is the ability to detect the variability? If ability is high, then we give a low number. If ability is low, then we'll give a high number. So based on that, we impact, probability, and detectability. Based on that, we'll assign three numbers, and by multiplying the three numbers, we'll give our RPN number. And based on the RPN number, if RPN number is high, if RPN number is high, then we will give that high severity to that variable, and that variable will be further investigate in the development part to establish a control strategy. So in detail, we will discuss these risk assessment tool in our SHQ9 guideline. But as of now, for the scope of this video, uh, these are the risk assessment tool which will help us to identify which is our critical material attribute or critical formulation variable or critical process parameter. So we define those critical process parameter or critical material attribute by using these tools. So friend, just this is a comparison between the two development approaches, that is minimal approach and the QBD approach. So the overall pharmaceutical development minimal approach is mainly empirical development research, often conducted one variable at a time. Means in the minimal approach, generally we study our variables relation with the CQA by using OFAT, one factor at a time. Like we are changing only binder, only disintegrant, or only one variable only at a time. So this is the generally done in the minimal approach. In the QBD approach, systematic relating mechanistic understanding of material attributes and process parameter to drug product CQAs, multivariate experiment to understand product and process, establishing of a design space, and PAT tool utilized. So in the QBD approach, we understand a mathematical relationship between the variables as well as the impact of variable on our drug product CQAs. Based on that mathematical relationship, we arrive on a design space, and we also use PAT tools so that our end product testing will be reduced. So this approach, this is the difference between the two development approaches, right? So then the manufacturing process. In the minimal approach, manufacturing process is fixed. Validation primarily based on the initial full scale batches, focus on the optimization and reproducibility. So but in the this QBD approach, adjustable within the design space. And if you have defined your design space, then within the design space there is a no change. So life scale approach to validation and ideally continuous process verification. Focus on control strategy and robustness, use of statistical process control methods. So these are the difference in the manufacturing process in the both type of development approach. In the QBD approach, we define a design space, and within design space there is no change. We ensure the our what is the our robustness of the process, and life cycle approach is used for the validating the process, and continuous process verification is involved in the QBD approach. Then process control, by the minimal approach, process is controlled by in process testing only, and based on the in process result, go and no go decision will come, and offline analysis is must in that. Whereas in the QBT approach we use pad tools. Based on the pad tools we get real time testing. Pad tool utilized with appropriate food forward and feedback controls. Process operation tracked and trended to support continual improvement efforts post approval. So, regarding product specification, primary means of control based on the best data available at the time of the registration. In the QBD approach, part of the overall quality control strategy, product specification is a part of overall quality control strategy based on desired product performance with relevant supportive data. So, control strategy for the minimal approach is drug product quality control primarily by intermediates in process material and product testing. Whereas control strategy in the QBD is drug product quality ensured by risk-based control strategy for well understood product and process quality control shifted upstream with the possibility of real-time release testing or reduce and end product testing. Then life cycle management, reactive problem solving and corrective action means the life cycle management during the commercial batches. So only kappa is used. Kappa is the solution for understanding the problem and giving a solution to the problem. But in the QBD approach, because you have a better understanding, you have a preventive action, you are continuously operating your process, and so because of that, there is a continual improvement facilitated. Dear friends, I am really thankful for your time. And in this video, we learned basics of the ICH Q8 only. This is only part one video. So in this, we learned basic of the QTP, basic of the CQA, basic of the CPP, and what is the difference between the minimal approach and the enhanced approach. In the second part of this video, we will understand what are the design space, how to define the control strategy, so that will understand in the second part of the video. So please must see that second part of also, and subscribe the channel. If you like the video, then please like and share the video. Thank you.